All right, welcome everybody. Today I'm going to be covering AP Biology topics 2.6, 2.7, and 2.9. 2.6 is on membrane transport. Your learning objective is to be able to describe the mechanisms that organisms use to maintain solute and water balance. Describe the mechanisms that organisms use to transport large molecules across the plasma membrane. Topic 2.7 is facilitated diffusion. Explain how the structure of a molecule affects its ability to pass through the plasma membrane. Topic 9 is mechanisms of transport. Describe the processes that allow ions and other molecules to move across the membranes. So first, our mechanisms of transport. Here are the three mechanisms that we'll then focus on and go more in depth through our notes today. The first is passive transport. Passive transport requires no energy, and molecules are going to be moved across the membrane either directly through the plasma membrane itself or through one of the integral proteins. Active transport requires energy. Look here in our image, we can see ATP being broken down into ADP, so releasing energy. Active transport will also use integral proteins. And then there's bulk transport. Bulk transport is going to require energy. That energy is used for the formation of vesicles that bring in large quantities of materials. So now let's look at these three in more detail. First, we have passive transport. In passive transport, the solutes are going to move with the concentration gradient, meaning they're going to go from high concentration of particles to low concentration of particles. We've got three specific types of passive transport. There is simple diffusion. We see this in things like carbon dioxide, where they go from a high concentration gradient right past all the phospholipids, because carbon dioxide is nonpolar, into our cell. Oxygen will also pass through simple diffusion. Next, we have facilitated diffusion. Facilitated diffusion uses transport proteins to help move around things like ions and polar molecules, because those have a hard time passing through the membrane. It also helps move some of our larger molecules. When it comes to facilitated diffusion, so we have two types of proteins involved. There is a channel protein. This is essentially just a pore or an opening that, as long as a molecule is small enough, things can pass through. Then there is the carrier protein. Here, things have to be a specific shape or have the correct charge to trigger a change in the shape of the protein that allows it to be moved across the cell membrane. But all of these still are going from high concentration to low concentration. Our last type, and not pictured here, is osmosis. Osmosis is the diffusion of water. And this occurs when a membrane is permeable to water, but not to the solute, not to whatever's dissolved in the water. Water will move from high water concentration to low water concentration. Oftentimes, water is going to use with the aid of an aquaporin, a special transport protein. Occasionally, some water, though, can pass through the lipid membrane. With our concentration gradients, things having high concentration or low concentration, we've got three terms used to describe those solutions and the concentration gradients of them. There's a hypotonic solution. Hypotonic has fewer solutes. So if we look at our model or example, our outside solution is 95% water and 5% solute. Inside the cell, which is the gray oval, it's 90% water, which means there's 10% solute. Percent should always add up to 100. Because of this, water will be moving. In this case, water moves into the cell, going from high water concentration, 95, to low water concentration, 90. If this is an animal cell, there is a chance that it can take on so much water that the cell membrane would break. Think like an overfilled water balloon. We call that lysis. In a plant cell, that cell wall is going to help them hold its shape, and we refer to it as turgid. That's the normal state for a plant. It needs that water pressure against the cell wall to help give the plant its structure and support to stay upright. Cells do have mechanisms to try to pump out of water actively if too much water is coming in. We see this in lots of our protists, like the paramecium has a contractile vacuole where they'll expel water using energy, but water will still passively move back in. Next, isotonic. This has an equal number of solutes in the solution and compared to the cell. So in this case, we have 90% water, 10% solute solution, and our cell is also 90% water. In this case, water will move equally because our molecules are always moving, move equally in and out. This is the normal or ideal state for an animal cell. A plant cell, though, we're going to describe it as flaccid. And this plant would start to look a little bit wilty. And if it was a plant in your house, it might have you going, that one might need some water. A hypertonic solution has more solutes than the cell. So here we have 85% water, 15% solute. Water goes from high water to low water. If it's an animal cell, it's going to shrivel up. Plant cells, we say, are plasmolized. The membrane here is pulling away from the cell wall. The cell wall, though, is still helping to maintain a structure. This would be a plant that looks like it's about to die if it does not get water soon. On to our active transport. In active transport, things are moving against the concentration gradient, or sometimes you'll hear the term up the concentration gradient. We are going from low to high concentration. It is going to require energy. Active transport is important in cells, particularly with maintaining what's called membrane potential, which is a topic we will talk in far more detail about when we get to photosynthesis and cellular respiration. With membrane potential, the different sides of the cell membrane, we've talked about how they're polar, but they're going to have different charges. The inside of the cell is more negative than the outside, favoring the influx or the movement in of cations, which are the positive ions. This creates a voltage gradient, so the cell is going to spend energy trying to get those extra positive ions back out. This change in electrochemical gradient is going to be very important with our reactions in photosynthesis and cellular respiration in driving our ATP formation. There's a couple of protein pumps that help with this process. We've got two main types, a sodium-potassium pump and then a proton pump, and that one's on the next slide. But here let's look at our sodium-potassium pump. With this one, initially the protein is going to bind 3 sodium molecules, and it also needs energy from ATP. That energy is transferred through the transfer of a phosphate. With that change, our protein changes shape, releasing the sodium out and bringing in 2 potassium ions into the cell. Because of this change, we now have 3 sodium outside, 2 sodium in, so this one is less positive or more negative on the inside of the membrane. This becomes very important with nerve transmission and your nervous system being able to function and send signals. Then we also have the proton pump, in which hydrogen ions, which are protons, get moved out of the cell. Protons have a positive charge. As they're removed, it makes the inside more negative, and this does require energy. This proton pump, we're going to see working with other proteins, other enzymes, in something called energy coupling. The energy from one reaction is used to power another. So in this case, our protein pump here uses ATP energy to move hydrogen ions out. Our proton pump moves hydrogen ions out. We now have a high concentration outside. The hydrogens want to diffuse back in. So as a hydrogen ion or proton diffuses in through this co-transporter, it can bring along with it other molecules. So this is an example of a sucrose co-transporter. Sucrose is otherwise too big to fit through the membrane. This picture, just to note, is not drawn to scale. A sucrose molecule is much, much larger than a hydrogen ion. This is just an illustration. Lastly then is our bulk transport. And we're going to look at two types. Bulk transport is any time we're moving large molecules either into or out of the cell. With exocytosis, there's going to be vesicles that have been made by the Golgi apparatus or Golgi body that are then brought to the cell membrane, where they then fuse with the membrane and release their particles outside of the cell. In freshman biology, we frequently talk about exocytosis being used for waste. It is not always used for waste. Some things like hormones and neurotransmitters will trigger a release and be released by the cell. So in our process of exocytosis, there's been some sort of protein made. It gets put into a vesicle by the Golgi apparatus, because that's the Golgi's job, is to package up proteins. Now we have our vesicle here traveling, releasing a signal molecule. That'll trigger a reaction in the neighboring cell next to it. This process of exocytosis becomes very important to our nerve cells and our endocrine cells that are sending out chemical messages. Messages are made inside, packaged by the Golgi, and then released through exocytosis. The reverse of exocytosis is endocytosis, so bringing things into the cell. With endocytosis, we're going to talk about three types. First, we have phagocytosis, referred to as cell eating, in which the membrane reaches out, so the cytoskeleton can manipulate the membrane. The membrane reaches out. A vesicle then is formed around a large solid object. This could be a piece of food. It could also be something like a bacteria. We see phagocytosis a lot in your white blood cells as they capture bacteria. This would then be combined with a lysosome that had digestive enzymes in it to break down the bacteria. We also have pinocytosis, which is called cellular drinking, in which they can bring in either large quantities of a liquid or large quantities of a small solute. Similar process in which there is vesicle formed by the pinching in of the membrane. These two, phagocytosis and pinocytosis, are non-specific. They'll bring in any type of particle. The third type is receptor-mediated endocytosis, which brings in specific substances. In this case, the substance is going to be a signal molecule, also called a ligand. And we'll talk a whole lot more about ligands when we get to cell communication. but a ligand is going to bind to a receptor molecule. So that's what these little Y things are. These receptor molecules can be some of the glycoproteins or the glycolipids or the carbohydrate chains that we talked about yesterday being on the outside of the cell membrane. So here our ligand is the little purple triangle molecule. Purple triangles bind with the receptors, signaling for a vesicle to be formed around them. So now these specific molecules get brought into the cell and then typically go on to trigger some other type of response.